Okay, well, let's continue. And I suspect I'm going to run out of time for. We may or may not get to launch into viruses today, but that's okay. We'll make up later. So I really wanted to cover, because you won't have the opportunity to experience these directly in the prep class, wanted to cover some of these useful diagnostic tools that you can use in the laboratory for detecting plant pathogens. So we very briefly talked about how you make antibodies that might specifically bind to and recognize our plant pathogens. We talked about monoclonals and polyclonals. But now I want to talk about how to actually use them. You know, what are the major assays? There are a number of them, but the important ones are things like ELISA. ELISA, or enzyme-linked immunosorbent assay. It's a very common, probably the most commonly used serological detection method for plant pathology and human pathology and a number of other things. I think they even have ELISA tests for HIV and whatever else. ELISA is really useful because it's rapid, it's very robust. You can you can have a range of skills and still get a good result. It's really useful for large-scale testing. You can do surveys quite easily and grind a whole bunch of samples. It's sensitive and it's accurate and semi-quantitative, even if I didn't spell it correctly. But it only detects what you look for. So how does it work? Do I have a picture? I do. We'll describe it here and then I'll show it in the picture next time. So it's an amplified reaction. So rather than, it uses that specific capacity of an antibody to bind to and recognize the pathogen, but it has an enzyme conjugated or bound to that antibody, and then we detect the enzyme. We detect the enzyme in an enzymatic reaction that changes colour. So the presence of the enzyme makes the colour go from clear to yellow, for example, and that obviously increases with time. So if the antibody is present with the enzyme attached, you'll get this yellow colour. So the only way the antibody is going to be present is if the pathogen is present. So you've got the pathogen, the antibody binds to it, the enzyme's there, you get that reaction, you get a colour change, and you get a detection. So let's see what that kind of looks like. Here's an example. This is what we call a double antibody sandwich ELISA. There's a whole bunch of different variants, but this is one of the most common ones. So we actually have little polystyrene microtiter plates, about 96 wells in each plate, so you can run about 100 samples per go. You coat the plate. This is usually you coat the plate with a capture antibody. So this is an antibody without an enzyme, an antibody that will recognise your pathogen. So coat the plate, and this is just going to increase your capacity to capture the virus or the pathogen that might be present at low concentrations. You then grind up your plant sample. So say this is, as I said, this is commonly used for virus detection. So say we've got a suspected virus-infected plant, and we suspect it's infected with cucumber mosaic virus. So we coat the plate with an antibody to cucumber mosaic virus. We then grind up our leaf samples in an appropriate buffer, and we add it to the well with the antibody there. So if the virus is present, cucumber mosaic virus, it'll bind to the antibody and get stuck there. If it's not present, or if it's a different virus, if it was turnip mosaic virus, it won't bind to that antibody because it doesn't recognise it. You then wash the plate. So that gets rid of everything except the stuff that's bound. So in this instance, where we've got our cucumber mosaic virus bound to it, the virus is still present. We've got rid of all the plant sap, we've got rid of all the green stuff, we've got rid of other potential pathogens that we weren't detecting for. We then add our antibody, but the antibody again, it might be exactly the same antibody, but this time it has got an enzyme conjugated to it. So an enzyme stuck to it. And it can only bind, because all the binding sites now are completely full, we've already saturated those on the plate, it can only bind if the virus is there. So the virus has been captured by this antibody, gets sandwiched by a second antibody with the enzyme attached. If there's no virus, the antibodies swim around, there's nothing to bind to. With our next washing stage, all of those disappear. So all of the enzyme is gone. When we add our enzyme, our substrate, enzyme substrate, in this instance, we get no colour change because there's no enzyme to catalyse the colour change. In this example, we've got enzyme present because it's sandwiching the virus, and it changes to a nice bright yellow. We've got a positive reaction. So, it's specific, it's quite sensitive because it's actually an enzyme reaction, so it actually increases with time. So even at very low concentrations, if you leave it long enough, that enzyme will continually catalyse that reaction, and you'll get a bright green, a bright yellow result. So it might take half an hour, it might take half a day, but you'll actually amplify the presence of that pathogen, and so it's quite sensitive. The negative, of course, maybe it is virus infected, but you picked the wrong antibody. So it's going to give you a negative result, but it's a negative result for the virus you tested for. If you don't know what the virus is, if you don't suspect what the virus is, you have to do multiple tests. Maybe you test for 3 or 4 or 5 or 6 different viruses to try and find out what's there. So as I said, that's very useful and it's really quite useful for broad-scale surveys. Say, okay, we know cucumber mosaic virus is a big problem in our lupin crops. We want to survey for that. You can coat plates with 100 test samples per plate. You can coat a couple of hundred plates. You can have people sitting there grinding plant samples with little mangles all day and collecting hundreds and hundreds of plant samples. Load them onto the plate, and you can test thousands of samples in a day or two, rather than staring down the microscope or electron microscope, which is going to take half an hour per sample. It's going to be much longer and much more difficult. So ELISA is really important, and particularly for virus samples, but can be used for other things too. Lateral flow devices, as I said, exactly the same technology as they use in pregnancy test kits, but they're now available for specific pathogens in agricultural systems. So pull it apart. How does it work? You've got obviously something that actually allows wicks through fluids through the actual device itself. You've got a pad here for loading the sample onto. You've actually got antibodies relative to your conjugated antibodies relative to your pathogen. You've got a test line and a control line. It's important to have both so you know the test's working. So the control line detects the antibodies themselves, I think. So it's always going to work out positive. So you've added antibodies, eventually it flows down, they're going to bind to these ones. So these are antibodies that recognise antibodies. And antibodies are proteins, so you can get antibodies, if that makes sense. These ones here are going to be specific for the virus. So you add your sample, it flows through, you get your antibody bound to the virus. It'll flow through. If the virus is present, you'll actually get a positive result on your line here, and also a positive result because it recognises the antibody. If the virus is not present, it'll pass this first line, you'll get no binding, but you'll get binding to the second line, so you'll get 1 or 2 bands. So this is really neat because it's essentially you can just take a leaf sample, squish it up, put a drop of sap on this in the field itself, and you get a result in 10 minutes or 5 minutes or whatever. Very rapid, quite sensitive, and something you can use in the field. So you don't have to take samples and shoot off back to the lab and then grind it up and 2 days later come back and tell the grower what the result is. You can go out there, rip one of these out of your pocket, grind it up, go, yep, you've definitely got cucumber mosaic virus because this has proven it. The symptoms look like it, now I've confirmed it. So very rapid, very easy to use, very robust, accurate. Once again, for the tested sample only, tested pathogen. It won't. It's specific for that pathogen. It won't tell you, hey, you've got a virus. It'll tell you you've got cucumber mosaic virus. It is, however, quite expensive. So you wouldn't be using this for large-scale surveys. Each one of these is probably a couple of bucks, or even more. So you're not going to be testing thousands of plant samples by this manner. It's also not good for large-scale testing, because even if you were, you really don't want 10,000 of these things laid out and grinding them up and spotting them all on. There's more efficient systems for that. It is specific for a single pathogen, and obviously for all serological assays, another limitation is you actually have to have an antibody. So mostly these days we buy them. There are companies that make antibodies, and you can search those companies and say, I want an antibody to cucumber mosaic virus, and they'll sell you one. But if you've got a brand new virus, if you've got a brand new agent, or one that's not that common, and there isn't a commercial antibody to it, you can't do any of these serological tests without an antibody. So you either have to make it yourself, which is not easy, and can be expensive and requires specific skills, or you use a different test. You can't do serology. What else can we use serology for? You can actually combine it with microscopy. In this case, this is with light microscopy, and this is with electron microscopy. So here we've got light microscopy, once again detecting viruses actually within a particular plant sample, but they've got an antibody connected to a fluorophore, so a fluorescent tag, and you've got your sample. There we go. We've got things that might have been viruses, but everything else was sort of going green. But we added this antibody with a red fluorophore on it, and then we put it under a different light system, and oh, these are glowing red. These are our virus, and that's exactly where they are. Rather than, here's some blobby things that might or might not be, we're not quite sure. With electron microscopy, they can do things like, well, one, they can improve its sensitivity. They can actually coat the electron grids, or the TEM grids, and actually capture more viruses on them, so you increase the concentration and increase the likelihood you're going to see it in the first place. But then they can also do things, and it's probably really hard to see from back there. I don't know, does these guys look a bit fuzzier than these guys to you? What they've done here, it's just absolutely crude, is you've got your virus prep, you've got these particles, and you've got virus 1 and virus 2, and they're all little isometric particles. They all look pretty much the same, except this is a different virus. So how do we know it's a different virus? Because we've used an antibody to this virus, and we just flooded the electron microscope grid with this antibody. And so millions and millions of proteins are bound onto the virus particle. All the antibodies are sort of bound onto it. So now when you look out into the microscope, instead of having a nice clean outside image, it's a bit fuzzy because it's just you're actually physically seeing the antibodies bound to the virus particle. So you might actually have a mixed example where you've actually got two viruses that look exactly the same under the microscope, but they are two different viruses. You add an antibody to one of them, and you see some fuzzy circles and some clean circles. You know you've got a mixed infection. So it's another way of seeing it, but also then identifying it by serology. So it can increase sensitivity of electron microscopy. In this instance, it can not only detect it, but can also tell you where in the cell it is. So if there's an interest to know what parts of the cell it's actually invading, this is localization as well as detection. Again, you need an antibody, and it's pretty low sensitivity compared to ELISA or lateral flows. So you'd use it for some specific reasons, but not routinely. It's also absolutely shocking for, you wouldn't be doing surveys doing this sort of approach. It's going to take you forever. So those are the main serological tests that we can use. Actually, for those who come back and do plant pathology, the advanced course next year, we will actually do an ELISA test. So we'll actually have a go at running one of those. There are also nucleic acid based tests that most organisms, well, pretty well all our plant pathogens we're dealing with, all the biotic ones, have a genome, have a nucleic acid genome, and they will be, the coding of that will be specific to the pathogen itself. So we can use detection tools that look for specific coding, specific nucleic acid codes that will actually tell us the presence of that pathogen. So we can use DNA fingerprinting. Now this is methods particularly used for organisms like fungi, some fungi where they can look at variants within species. So that's particularly useful if you're doing, say, epidemiological studies. So maybe rather than detecting the presence of a pathogen, you want to detect strains of the pathogen. You want to look at variants of the pathogen, and maybe that's related to how it's reproducing in the field, how it's crossing with other strains of the pathogen, but also where it's coming from. So maybe you've got a strain A in this part of the field, strain B in that part of the field. As the disease progresses, which is the most aggressive strain, which is the one that's actually dominating? Maybe one of them is going to be fungicide resistant and one of them is not. So it helps you understand population dynamics. Generally less used for detection and diagnosis and more for those sorts of differentiation. But there's a number of tests, and we're not going to go through detail really how these run, but just to be aware that these exist. RFLPs, restriction fragment length polymorphisms, these rely on enzymes that chop nucleic acid at specific sites. So we all know our nucleic acids have different codes, so they might be A, T, G, C, A, C, C, blah, 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 blah. That's your nucleic acid coding. These enzymes, and there's a whole bunch of different enzymes, will specifically recognise a certain string of maybe 6 or 8 nucleotides, and it recognises that and then chops it at a specific point. So every time that appears, so if you isolate some DNA and every time that appears, it'll chop it at that point, and then you'll get a cut. And then you can actually look for the size fragments of that. So if you've got obviously one big molecule and it cuts here, you end up with a short one and a big one. If you've got one that cuts there and cuts there as well, you might end up with a short one, a slightly bigger one, and then a bigger one still. So the number of cuts and where they cut will depend on, will give you the number of bands you get, the number of different size fragments, and what size they are. And you can use that to actually identify things based on the presence of these motifs, these cutting sites. It's not the most sensitive assay, and it's pretty tricky to do, but that's the way that works. Others use an amplification process, so they actually, rather than just having to isolate large amounts of DNA and chop it up, they amplify the DNA and chop it up, called a rapid marker. And other ones use microsatellites, which are, again, pieces of sequence in a genome, usually repeat. So they might have a piece where they've got lots of 5 ACs in a row. And if these can be identified, and they tend to vary between individuals. Sometimes it might be 5 copies, sometimes it might be 7 copies, sometimes it might be 3 copies. But once these have been identified, you can actually design tests that will amplify and detect that particular region, and then you can determine, have I got 3 of them, have I got 5 of them, have I got 7 of them, and the number of repeats will be specific, will fingerprint that particular strain of the pathogen you're dealing with. So these microsatellites can be useful. Once again, you just look at the size of those repeats, and use that to actually determine which strain you're dealing with. Let's move on. By far more important for diagnosis and detection is a test called polymerase chain reaction, or PCR. Who's heard of PCR? Who's not heard of PCR? Right, okay. So this PCR is a really interesting technology. It's bringing molecular biology to non-molecular biologists. Pretty well anyone can do it. It's kind of a piece of cake. It was actually, I'll tell you a story, it was actually discovered by this guy, Kerry Mullins, I think it is, in California. So he was looking at ways to amplify nucleic acid. And the way it kind of works, the way nucleic acid is amplified, or the way it normally is reproduced, is you've got your nucleic acid with all your different codes. You get a short piece of nucleic acid that is homologous to one end. You add an enzyme, and the enzyme copies over and makes, so if that's an A, we'll put a T there. If that's a T, that'll be an A, C, and that'll be a G, etc. We'll copy along and make a copy. So if you want to copy nucleic acid, you have a little, what they call a primer, a short piece of nucleic acid that'll recognise this bit, and an enzyme. And the enzyme copies it along. So he was going, well, if I do that, I'll just make one copy. If I then go at this end, I can make a copy backward that way. But if I use both of them, I can just keep copying, and end up going, that piece between there will increase logarithmically, and you'll end up with millions and millions of copies of that specific piece of nucleic acid. And that can be really useful in terms of identifying things as well as working out variation. The issue with that was every time you ran the reaction, you had to kind of add more enzyme, and it was because you need to heat it up, split it apart, and you need to cool it down to let it join back again, and then let the enzyme work. So this guy was struggling away, he couldn't get this thing to work, and then he was shooting off down the coast on having a weekend off, and drives past, and I think no, it can't be. No, it was California, so it wasn't in Hawaii. So he didn't actually see a volcano, but it struck to him that these volcanoes are obviously hot, and you have these microbes, you have these bacteria that live in these very high hot conditions. And he's going, what I need is an enzyme that doesn't die when I heat it up to 90 degrees. Because every time I heat it up to 90 degrees, the enzyme died and you had to add a new enzyme. What if I go and get these bacteria that live in volcanoes and isolate their enzyme, their DNA replicating enzyme, and then I can do my temperature cycling and the enzyme won't die. And that's exactly what he did, and he rushed back to the, he's having his weekend off, didn't eventuate, he rushed back to the lab, he isolated this protein from his volcano bacteria, and it works, and got a Nobel Prize for it, so it's probably worth it. Anyway, so essentially that's how it works. You have these two short pieces of DNA that recognise your target, one going forwards, one going reverse, and an enzyme, a thermal stable enzyme. And you've got these different phases. First, denaturation, because we're dealing with all our organisms are double-stranded DNA. So you can't copy it until you pull it apart. So you heat it up to 95 degrees and they fall apart. So you end up with your strands are single-stranded. And it also straightens them out because we keep drawing them as these long straight molecules, but they're not, they're all twisted up. So you heat them up, they straighten out, they separate, you end up with these single-stranded molecules. We then have an annealing temperature, so we drop the temperature down and these primers start binding on at the appropriate sites where they can recognise it. We then let the enzyme do its job and stitch up the bits between them. And that's kind of diagrammatically what it looks like. So we start off with our target, we We put a primer on it, we make one copy. We then blow it apart with a second primer and keep going, and eventually you end up with millions and millions of copies of the piece of DNA between those two primers. So we know the length of that piece of DNA, what it should be, because it's between where this one binds and when this one binds. So that might be, 500 nucleotide space. We know exactly what it is because we know what the genome we're looking for, and these primers are designed for that. Then we can run a gel and look for a presence of this of the right size. So here we go, we've run a gel, and here we've got big bands, so that's probably about 1,000 base pairs. These ones here are probably about 500 base pairs. These ones here are probably about 200 base pairs. So if we, for diagnosis, we design our primers based on the genome of our target organism. So we pick primers that are specific for our target organism. What makes it even more specific is it's not just one primer, the two primers have to be there, and the two primers have to be exactly this far apart, because if we're looking for a band that's 300 and we get one that's 500, we go, that's not right. We know that's not it. We've got a band, but it's not the right band. We wanted one where they're that far apart and they're going to produce the band that we expect. So from a diagnostic perspective, this is very, very sensitive. You get a logarithmic production of this, very, very high sensitivity, and quite specific because it's based on that DNA homology between the primers. Not only the homology of the primers, the fact they can bind, but also how close they are to each other, and you can specify that. Any questions about how that works? That's PCR in 2 minutes, which is a bit pushing, but anyway. If you got that works with DNA and DNA only. So if you've got an RNA virus, so most plant viruses are RNA genomes, they're not DNA genomes. So that's problematic. If you want to use PCR for detecting an RNA virus, you can do something else first. So what you do here is use another enzyme called reverse transcriptase, and this enzyme turns RNA to DNA. So it's just one extra step. So you isolate your RNA from your virus-infected plant, you treat it with this enzyme, reverse transcriptase, and it makes a DNA copy of your RNA, all the RNA in that cell. You then use your DNA, use primers to the DNA version of your virus, and then run through the PCR process exactly as before. And if you get a nice band, then you've got a positive detection of your RNA virus. Of course, you've just made DNA version of it. And there's more variants, and this is becoming more critical, more important these days in diagnosis. There is something called quantitative or qPCR, sometimes called real-time PCR. So it's exactly the same process of heating up, cooling down, heating up, cooling down, breaking apart, annealing, extension. Except detection this time is, instead of producing a bunch of DNA of set size and running a gel and looking for a band in the gel that is of 300 base pairs, this one instead uses what we call fluorophores, dye labels, on either the primer or on part of the building blocks that make the stitch up the bit that you're making. And these fluorophores are then detected by their fluorescence by spectrophotometry, really. So you've got a spectro busy analysing and looking for these fluorescent tags. And there's two basic systems, and you don't really need to know the difference, but just know that they are. One called TACMAN, which is they actually use 3 primers. So you've got your target DNA, you've got 1 primer in that direction, one primer in that direction. That's doing your PCR. Then it's got a third primer with a label on it. So what it's doing is make that product, and when the product's present, this one will bind and it'll fluoresce and go, yep, it's present. Don't make the product, it doesn't bind, you get no fluorescence and no detection. The cyber green ones, and this one here instead, you've got your primers and all these building blocks. It might be you fluorescently label your A, and so every time an A goes in, gets built on there, you're adding a little fluorescent tag, and then you detect the fluorescence as you're building that PCR thing. So as you're multiplying lots of it, you end up with the capacity to detect what you've made with a fluorescent dye. Why do we do this? It's very sensitive. About 10 times more sensitive than ELISA, which is about, than PCR, which is about 10 times more sensitive than ELISA, which is probably about 100 times more sensitive than isolation. Very, very sensitive. Very useful for detecting very low levels of what you're looking for. What can be valuable about that? It might be really good for, it tends to be moderately expensive to run, but it might be, I've got 1,000 plants, I know I've done it myself, ground up 1,000 plants into one tube, do a single test, positive result. It's that 1,000 plants contains at least one infected plant. So the sensitivity is such that you can do that. Was it someone was saying, even with standard PCR, they were sort of saying that the theoretical detection is that if you drop one molecule of DNA into an Olympic-sized swimming pool, we should be able to detect it. It's very, very sensitive, and this is 10 times that. In theory, I wouldn't trust you to be able to do that, but that is the theoretical detection possibility. Where am I at? So, what are the pros and cons? Sensitive to extreme sensitivity. So, if you've got very low target, which is hard to find, it also allows you to bulk samples, so you can test. Testing 1,000 samples might not be that useful because you've then got A positive and you still don't know if it was 1 in 1,000 or 1,000 out of 1,000. But at least if you are looking for rare things, or even looking for quarantinable pathogens where you don't expect to find it, but you need to test a hell of a lot of samples to make sure it's not there, rather than testing 10,000 individual samples, you can test maybe 10 lots of 1,000 and then get yep, it's clear. So seed certification, well, in fact that was used commercially in Western Australia. That cucumber mosaic virus in lupin seed is a real problem. It's in lupin crops is a real problem. So they want to make sure the seed is free of the virus. So they actually do both ELISA testing but also PCR, where they grind up 1,000 seeds, run it through a single PCR, and if it's negative, then they go, okay, we certify your seed because less than 1 in 1,000 positives. So quite sensitive and quite useful. The other advantage is, because you're actually producing DNA in that, between those two primers, you can take that and actually do things with it later. You can actually sequence it and actually work out the sequence and confirm that it really is what you're dealing with. So if you're detecting a bacteria and you've got a band and it's of the right size, and you're still not sure, you want to absolutely make sure it's what you're dealing with or it's what you're really looking for, you can then take that piece of DNA, send it off somewhere and get it sequenced, and then look at the A's, T's and G's and say, yep, that's actually exactly what I expected it to be, and it's not some spurious amplification that wasn't right. You can automate it. They've got robot systems now where you get your samples, you pop them into bags and you press a button and it kind of squishes them all and extracts it all and runs the whole assay and gives you results at the end. So again, high throughput, works really, really well. Negatives, again, it's specific for a single pathogen. You're testing based on the presence of these primers. So if you've got an unknown sample, you're once again, you're saying, I'm looking for cucumber mosaic virus, I'm using primes to cucumber mosaic virus. If something else is present, you won't detect it. You'll only detect what you're looking for, exactly the same as serology. because it's super sensitive, you've got a potential issue of contamination. So if you are a lab that's doing routine testing for cucumber mosaic virus in lupin seed, for example, and you're grinding up lots and lots of lupin seed, and there's lots of lupin seed dust all over the room, there's probably lots of virus particles floating in the air, and then you get a healthy sample and you run it through there. If you're not careful, some of those will settle into your sample and you get a positive result. So contamination can be a real issue. When PCR was first set up, they were doing lots of, I remember hearing, at least from some labs, they were actually doing work on, I think it was Y chromosome stuff, and they refused to employ males because they were contaminating all their samples all the time. Just by handling it, even while wearing gloves and things. I think procedures are a lot better now, so you don't need to do that sort of stuff. But the potential for contamination is such that some people get quite paranoid. I remember some labs where they go and do their extractions in a different building and then bring it back there and do the amplification, because once you go to the amplification process, you're producing billions of copies of your target. That is your major source of contamination. So if you do extraction in a completely different room or different building, less likelihood of contamination. You've got to know some genome sequence. If it's a completely unknown organism, how are you going to design those primers? How are you going to make them in the first place? You've got to know DNA sequence. So it's fine if it's a defined pathogen, and you can. It's much easier than serology. You don't have to inject any rabbits. You just go and look in the databases, find some DNA, order some primers from a company that makes them. Dead easy. But you've got to have that information to start with. And it can be costly. And it can be heart destroying too. What else can we do? There is other assays. This assay is relatively new, been going for a decade or so. Again, it amplifies DNA. It's called LAMP. And what does LAMP stand for? Oh, I've forgotten. Whoops. I'll tell you next time. It amplifies DNA from an organism. The advantage of LAMP is that it doesn't need to go up and down in temperature. So it doesn't need to blast the at 95 to separate things and then cool it down and heat and cool and heat and cool and heat and cool. It can run at a constant temperature. It uses, and it's kind of hard to get your head around, so don't worry too much about the details, but instead of 2 primers that go between 2 sets, it's actually got at least 4 primers or sometimes 6 primers that actually bind on and then form a loop structure. And then eventually the enzymatic reaction is, rather than blowing apart double-stranded DNA to single-stranded DNA, linear and anneal again, it's actually using what we call a rolling circle amplification. So once you've got a loop, the enzyme can start ticking around here and copying it, and then that can fly off, and you do another copy and another copy and another copy and another copy. So just go round and round and round in circles, just continually copying and making lots of copies of your target and huge amounts of DNA in the process. So this looping process can occur at a standard temperature.
Yep. Does it just stand for loop amplification?
Yes, I'm sure it does. Loop amplified. What's the M? No, can't think what the M stands for. That's a polymerase, but I'm trying to think what the M stands for anyway. The detection, it can be done really crudely, and where this was initially used, like PCR is probably more sensitive, although this is pretty good. This is really useful. In fact, they designed LAMP systems for use in the field. So you can actually go there, you can add your sample, you let the enzyme chug around. You don't need an expensive machine that can heat up and cool down, because it's all done at a constant temperature. And they're measuring it by turbidity. It produces so much DNA, the solution goes a bit cloudy because there's so much DNA present. And the magnesium pyrophosphate precipitate. rather than the DNA really. And it can be measured, or you can add fluorophores to it and measure it by fluorescence, a bit like the quantitative PCR. So the pros are, it's quite simple, it's quite rugged, it doesn't need thermocyclers, it doesn't need temperature changes. It can be used in the field. Saying that, they've actually got qPCR machines, these ones that do fluctuate, that you can trundle off into the field and stick samples in and work as well these days. It's an enzymatic reaction. All these are enzymatic reactions, so enzyme inhibitors can be problems. But in this instance, this tends to be a bit more rugged and a bit more robust. The cons, you've got to make a hell of a lot more primers, so primer design is a bit trickier. Instead of finding 2 that work for all of the strains of the pathogen you need, you've got to find at least 4 or maybe 6. Multiplexing, I haven't talked about that. Multiplexing is where, again, you want efficiencies. As I said, all of these reactions, you're testing a single pathogen, like cucumber mosaic virus. What if you want to detect cucumber mosaic virus and tobacco rotavirus and tomato spotted wilt virus and something else? So you run 4 different tests, or with PCR, in some instances at least, you can load up 4 different sets of primers in the same reaction. So you've got tomato spotted wilt primers, and we've got our cucumber mosaic virus primers, and we've got our turnip yellow virus primers. We add them in the one tube. This one's going to make a product that's going to be 300 base pairs. This one's going to be 500. This one's going to be 1,000. We run the assay. They're binding on all specific sites. They run up, and we look for the band size. So if we get a band of 1,000 or a band of 300, we know we've got those 2 viruses, but not the other one. So that's called multiplexing. We have multiple testing in the one tube. So that, again, adds efficiency, reduces the cost, allows you to test more things. Not that easy with LAMP because of the number of primers you need. Any questions? Everyone blown away by molecular biology yet? The last, and certainly not the least, is genomic sequencing. Now this, again, is relatively new. It's not that new, but it's relatively new, where we have the capacity to sequence whole genomes. So I don't know if you remember back, goodness me, was it 20? At least 20 years ago, I suspect, when the first human genome was generated. That took them, was it 10, 15 years to generate that genome at multi-million dollars cost to generate the genome, with labs from, I think 4 or 5, probably at least 20 labs around the world, all working together to try and slowly stitch together and sequence this whole genome, and eventually they did it, and that was wonderful. Nowadays, you can get a human genome in half an hour, or a day at least, no more than a day, and for less than $1,000 by anyone. Or essentially, you don't do it, you just send it off to Korea or Japan or China or someone else, and they stick it in a machine and it goes bloop, and then they give you this data back. The issue these days is data overload. So these what they call next generation sequencing technologies, this has revolutionized the process. So this has caused where you get massive parallel sequencing, and you pump in a sample and it'll sequence the whole lot and give you huge amounts of information that then you have to try and deal with. Talking about many gigabytes of data. So, what do we do with it? Why is that useful? Well, nowadays, particularly for small organisms, it's a bit harder for fungi, but certainly for viruses and even for bacteria, getting a whole genome is now relatively easy. You've got a culture, or an infected plant, you can go and extract nucleic acid, send it off, blast the whole thing, get a sequence, and then look for the presence of a particular pathogen by the sequence. And rather than PCR where you're amplifying a couple hundred base pairs, or that restriction digest where you're really detecting 6 nucleotides, here you're getting the entire genome of the organism. So you can work out not only you've got it, but you can also work out all the variants that you do have, because there might be sequence variation. You can work out exactly what you've got because you're getting the entire genome information. Increasingly, for diagnostic purposes, this is becoming particularly useful for virus work because, as I was saying before, the major tests for viruses are things like ELISA, things like PCR, where you've got to know where they're good, but they only test what you're looking for. So, looking for cucumber mosaic virus, we put in a cucumber mosaic virus antibody, and we work out if it's there or not. But what if we've got a sample, we have no idea what the virus, we're pretty sure it's a virus, but we have no idea what the virus is. We test as many antibodies as we can, and we're still getting negatives, we still don't know what it is. How do we find out what we've got? The old days, you'd try and do some just crude purifications and hopefully isolate something and then try and characterize it. Nowadays, you take your sample, you grind it up, you send it off to Korea, and they pump out a huge amount of, you extract the RNA, they turn it into DNA, they sequence the whole lot, they send it back to you, and then you search through that mountain of information, most of which will be plant stuff, but you can filter all the plant stuff away and look for virus sequences. And then, boom, they'll pop out, you'll get the whole genome, and you'll go, here's a brand new virus no one's ever discovered before, and I've got the whole genome of it, and it's causing that symptom. So we're getting, if you look at the literature now, we're getting hundreds and hundreds of papers coming out now where people are discovering brand new viruses because they're testing samples and these genomes are popping out that no one ever knew to look for before, but they're finding them. That can be really useful, again, for a sample that you have know nothing about. You don't need to know primers, you don't need to have antibodies. It can be no information whatsoever, and you'll get the result. It can also be valuable for doing large-scale surveys. I've seen examples where someone's gone, tree crops are particularly bad for viruses because they're perennial and they can accumulate them over time, and they tend to be vegetatively propagated, so they can be problematic. So they went to an orchard and they picked up, leaves off of a couple of hundred plants. They combined them all together, sent them all away. 20 viruses appear, and you go, okay, now I know what to look for. Then you can maybe devise more routine assays like PCR, and then screen and find out which trees got which, and so on. But it can be really quite useful as a tool for identifying things you don't know about. Detects everything. If there's 2 viruses, it'll detect both of them. You don't need to know any genomic knowledge. It gives you the full genome sequence to allow identification. Currently, it's still pretty pricey. It might cost you anywhere between $1,000 to $5,000 to do a sample. So if you're testing, if you're worried about one plant, unless it's your mum's favourite pot plant and it's worth, the $5,000 is worth investing in because, but you wouldn't do that routinely for lots of individual plants. But there might be opportunities where you can bulk test, or if you've got a brand new disease and no one actually knows what it is, this is the way to kind of find out. Slightly less sensitive than qPCR, and you've got to have good computers and good software to actually manage this ridiculous data set you get. Okay, so I have successfully killed the virology lecture. That's okay, we'll do that next time. But to summarise some of these lab tests, don't underestimate looking at the plant to start with. Look at the plant both macroscopically, microscopically, try and find presence of a possible pathogen. That'll at least give you the clue what to try and isolate it from, what to do, or what subsequent test to use. For many fungi, those sporulating structures might be present on the plant, but not on your isolated pathogen. Other than that, there are many other tools for detection of plant samples, either in the plants or in the environment. You can isolate them for further characterization, or you can use some of these other tools. And this is, you can look at that later too, but it's sort of my crude estimation of some of the pros and the cons or otherwise of some of these tests. So, symptomology, well that's good, you don't need to know anything about it, you're just looking at symptoms, so there's no need for any pathogen-specific information. But same with microscopy, you're looking for whatever's present, even electron microscopy, whatever's there. ELISA, well yes, you need an antibody. Lateral flows, PCR, all of these you actually need to know what you're dealing with, you need to have some information. Genomic sequencing, no, you can do that without any knowledge at all. Sensitivity from pretty crap through to incredibly good. Robustness, some of them like ELISA, really good. Some of them like genomic sequencing, electron microscopy, it can be a bit flaky. So you can go through that list too and actually look at some of the advantages. Again, how good do you need to be? If you're relying on symptomology, it's relying on being an expert and understanding what you're dealing with. So pretty high level of expertise to interpret symptomology as a diagnostic tool. Lateral flow device, goodness me, all you need to do is stick the sample in and be able to read one stripe or two stripes. So no real knowledge needed to run that test. To interpret the test, maybe. What does it mean for the person? I've got a positive now. But to actually run the test, pretty low. And even things like genomic sequencing, I've probably underdone it there. To actually do the sequencing, because you send it away and someone else does it for you. To interpret some of the data can be a bit tricky. Expense, how quickly you can get results straight away, ones that take a while. And the value in terms of being able to identify what you're dealing with. Things like genomic sequencing, incredibly good. I put microscopy as pretty damn good because you do actually get to see it, and particularly if, like a downy or a powdery mildew, if you observe an infected plant, you see that structure, you know what you're dealing with. You don't need to do any of these other tests. It's already there, you know exactly what you're dealing with. Oh, oops, okay. I thought I'd finish. 5 more minutes, that's fine. The last thing we're going to do, and this is actually pretty important, so we will make sure we do this. Cox postulates. Anyone heard of Cox postulates? One person, that's good. Yeah, okay. So this guy, Robert Koch, he was actually a doctor. Not a scientist, he was a doctor, but he had a fascination. This is when microscopes first came out, and he was a sort of a backyard scientist, and he was particularly interested in anthrax. So there was anthrax epidemics and killing off cattle, which of course is also a potential issue for people. So whenever a cattle died, they quickly buried it and tried to get rid of it so it didn't start killing people. So he started looking at using his early microscopes and started looking at the causal agent of anthrax and seeing, okay, look, you see these wiggly bacteria around there. And he started to study them and but he wanted to characterize them, and he wanted to prove that these things he was seeing were the cause of anthrax. So what he did, which was absolutely novel, was he said, I've got to get it out of the diseased animal and study it, and then I've got to put it back into a healthy animal and generate disease. That's the only way I can prove it. So he actually used, they didn't have agar plates back then, he used actually eyeballs. So a nice healthy cow, go to the abattoir, wrapped up some eyeballs. You get his bacteria from blood from an infected animal, and he'd inject it into the aqueous bit of an eyeball, and the bacteria would grow like crazy. And that's quite clever because that's actually fairly sterile. So he created a sterile medium. He grew up his bacteria. He studied it and says, well, that looks like the same stuff I saw before. He then took that and injected it back into another animal, and it died of anthrax. And he went, oh, wow, that's interesting. He then went and visited some of the scientists who were puzzling over the same thing at the moment, and they almost fell at his feet thinking, my God, you've actually solved our issue that we could never... We were trying to prove that these bacteria were the causal agent, and you've done it. So he beat the scientists to it, and got the Nobel Prize for it too. So what does it do? How do you do it? First, you need to examine your plants, and it needs to have, the pathogen needs to be consistently associated with the plant. So if you've got diseased plants without the pathogen, it obviously is not the pathogen. You need to isolate it into pure culture and characterize it. You then need to inoculate it to a healthy plant, and it needs to induce the same symptom that you first observed. So it needs to cause that disease. Then you need to isolate it once more and make sure that this new diseased plant actually still has this new pathogen that you've got. That is Koch's postulates. And if you can complete that, you've fulfilled the best proof of cause for a particular pathogen. 2 minutes. What are the problems? Quick.
Not all can be onto an agar plate.
Exactly. Obligate pathogens, rusts, smuts, viruses can't be isolated. So they've made modified Koch's postulates where you kind of semi-isolate them. So you take them out of one plant, you put them into another plant, and then back again. It's not quite the same, but best they can do. This is very pathogen focused. It works perfectly well for anthrax in cattle, not so well if you're dealing with those interactions with environment and time. So the disease pyramid, we can't forget that. So if you've isolated a pathogen onto an agar plate and then you stick it back onto the host, but you don't replicate the environmental conditions, you might get no infection, might get no disease. You haven't fulfilled Koch's postulates, but it might still be the pathogen. So understanding that whole system becomes pretty important. And similarly, how do you inoculate? It might be a pathogen that can only invade as a seedling, and then you try and inoculate an adult plant. Not going to work. It might be that it needs to actually have a wound site in the root, and you sprinkle it on the surface of the leaf. So actually understanding the biology of that interaction is also important. Right, so it's really important. If you can prove Koch's postulates, then you've proven that your new plant pathogen is a plant pathogen. If you can't, there might be a number of reasons why not, and it doesn't necessarily mean it isn't. Thank you for that. See you after the break.